Hello, friends. I'm here to share with all of you the basic science about the current pandemics, which has kept the whole world under the swings. One of the greatest difficulties that we are facing nowadays is lacking of knowledge. It's about the lack of clarity, what's going on, and what lies ahead. We, as students or scholars, especially feel a need to deepen our knowledge about the pandemics or about the situation. So the pandemic or the situation, you know it very well. So it is COVID. So I'm talking about the COVID-19. So so the COVID-19, the whole scenario came into notice in December 2019 and COVID-19. So in December 2019, a series of cases of pneumonia of unknown origin were reported in Wuhan, the capital city of Hubei province in China. In January 2020, the causative agent who caused this pneumonia-like cases has been isolated and characterized. That is a virus, and in 12th of January 2020, WHO, the World Health Organization, has tentatively named the virus as COVID-19. That is, N means N is novel, CO is corona, VI is virus, D is disease, and 19 is 2019. In 30 January 2020, WHO issued a public health emergency of international concern. That is, on 30th of January, WHO has issued a PHEIC, public health emergency of international concern. On 11th of Feb 2020, WHO, that means WHO, formally named the disease caused by this and COVID as COVID-19. That's coronavirus disease 2019. But at that time, based on its genetic relatedness to known coronaviruses at that time and established a classification system, the International Committee on Taxonomy of Viruses, they have classified and named this code as 219nCoV as SARS-CoV-2. Why SARS? We'll see ahead of this topic. On 11th of March 2020, the same organization, that's World Health Organization, formally characterized the global spread of this COVID-19 as a pandemic. So COVID-19 has been declared as pandemic in 11th of March 2020. And as the first to be caused by as coronavirus. Now, what is this coronavirus? So we'll see about the classification of coronavirus. So how coronavirus has started, where from it came, and its activity. It is genetics most importantly, and the diagnosis and the clinical manifestations, the management, and importantly ethics of the same disease. So how we should deal with the patients, how we should deal with the controls, how we should deal with this disease according to the ethical committee. What are the recommendations from the ethical committees? So first of all, what we'll see is this the virus, what it actually is. So we can say the coronavirus. We'll go into deep knowledge of this coronavirus. First we will see what it is. So the coronavirus is positive sense. It is a single strand. That means the genetic material of this coronavirus, like we have the genetic material of so the genetic material of this coronavirus is single stranded RNA So it's called RNA virus, as genetic material is RNA. So we can say this is RNA viruses with helical capsids. That's a protein. So after that, so this helical capsids, so it is known to infect various hosts like humans, bats, and like other mammals, birds, etc. So the thing here most importantly is like this coronavirus is importantly belongs to, if we are going to, so we are looking for the classification, that is we are looking for classification from which family this coronavirus has came and to which family it belongs. So this coronavirus belongs to the order of nidovirales. This nidovirales has a family of Coronaviridae. Coronaviridae has further been classified into two types. First is coronavirinae and torovirinae. Coronavirinae and torovirinae. This coronavirinae has further classified into genres as alpha, beta, and gamma and delta. So this alpha coronavirus and beta coronaviruses are known to infect humans specifically. Out of this, there are some strains which are less pathogenic, like in alpha coronavirus there are some human coronavirus 229E, human coronavirus NL63, and some others also like HKU1 and OC43. These are also contributing, but these have mild pathogenicity, so these contribute to only seasonal cases of common cold, like fever sometimes, but sometimes these are linked to severe pathogenicity, or we can directly say respiratory illness, like SARS or MERS. SARS is severe acute respiratory syndrome and MERS is Middle East respiratory syndrome. Right. So in beta also, beta are known to cause severe SARS-CoV, that's severe acute respiratory syndrome coronavirus, and this is responsible for SARS worldwide, this beta coronavirus outbreak, which has been reported in 2002 and 2003, and MERS also by MERS coronavirus. Alright, so this was all about the classification of this. So we are not talking about the Toronoviridae. Toronoviridae, if we are talking about, there are also the two types of this coronavirus has also been, this Toronoviridae has also been reported, that is Toronavirus and Bapinivirus. Toronavirus and Bapinivirus. But our major concern is coronavirus. So the types of the genera of coronavirus that are mainly infecting the humans and other mammals, particularly we're talking about here in the humans. So alpha coronavirus and beta coronavirus. Alpha and beta are most commonly contributing to human pathogenicity, and beta severely affects the humans through two reported disease, which are SARS and MERS. But alpha coronavirus is reported with low pathogenicity, that's human coronavirus 229E, human coronavirus coronavirus. These are the strains of alpha coronavirus. These are usually cause the seasonal cases of common cold, like but seasonal cases of common cold are like fever, and sometimes the running nose, and sometimes the blocked nose, and sometimes the sore throat. But these are also sometimes linked to severe respiratory illness like SARS and MERS. But most importantly, the majorly for SARS and MERS, beta coronaviruses are responsible. Now we will see the genome of genome means the genetic material of coronavirus. Coronavirus has the largest 30 kb size of RNA, that is single strand. Okay, but this is it has the largest, so this 30 kb is largest genome of all RNA viruses, right, which is infecting the humans. So this first point. Second point, we can say it has, from the genetics point of view, it has non-segmented genetic material, is non-segmented with 14 ORFs, that's open reading frames. Third, if we see the closest links to this virus, the closest identified related to SARS-CoV-2, that's COVID-19, is a virus that has been isolated in bats. The virus which has been isolated in bats and the SARS-CoV virus, SARS-CoV-2, the relatedness is 96% identity in sequence. That is, sequence identity is 96%. That is, they are more or less they are almost, we can say, near to 100%. They are identical. The fourth one is here. Like these are the home take points. So this is from where it started, that is, all of you might know this starts from the live animal or seafood market in Wuhan City in China, but it has also been found in that market, in that animal market, where the civets are pigs. Were being eaten by the people. So it has been found that it's actually being supposed that the intermediate hosts between bats and us, that means humans, maybe the civets are pigs. Now, if we will see the virus structure in detail, that is how this virus looks like. The virus looks like, if we'll see, this is a club-shaped virus. That is club-shaped. 2-3 identities are there. Like first one is this club-shaped. It has trimeric surface protein, which is called spike glycoprotein. This spike glycoprotein gives this virion a crown-like appearance. So this crown-like appearance of this virion, so hence this virus is called coronavirus. Corona is actually a Latin word, right? So second point, we can say this coronavirus contains 4 major structural proteins. First one is spike protein. Second is envelope protein. Third is membrane protein, or we can say matrix protein. And fourth one is nucleocapsid protein, right? So if we see this as how important these four structures of proteins are, here we write this spike protein, for example. Spike proteins. This spike protein, if we'll go through the structure, or we'll make like the structure of this virus. So the structure of this virus looks like this. This is the circle, and this is another circle. And see, I told you first is spike protein. So this spike protein is this spike protein actually. So this spike protein, if we will see, this spike protein has a trimeric structure, and in this case this is the membrane. This is a membrane, and this is like, we can say, this is these two and this three. So this is trimeric, that is 3 domains are there, 1 2 3, 3 domains in across this whole membrane. There are the spike proteins on this membrane everywhere. For example, this, and here we can say this is spike protein. Yes. After that, I told you there are envelope proteins. Envelope proteins are also on the membrane. So that means like a smaller structure here grows like this. This envelopes. Why it's called the envelope protein? Because it envelopes the nucleocapsid. It envelopes the core genetic material. So this is called envelope protein. And third one is matrix. I told you, our membrane protein. So membrane protein is also like on the membrane dimeric structure. This is trimeric structure. That is transmembrane actually. Transmembrane means on the membrane, in and out of the membrane. So this is called membrane or matrix protein. Sometimes one more protein in some strains of the coronavirus, that is HE protein and hemagglutinin esterase, and that is also like the, if we can see here, that is found in some strains. Only some strains it hasn't found till yet, not in all coronaviruses. So it's shaped in red. So this is called hemagglutinin esterase protein. Now if we go through this, so this is the structure. Now this spike protein, that means this one, this spike protein binds with the host. This spike protein, it binds with the host cell receptors to facilitate the entry into the host cell. So that means this spike protein, for example, this is human cell, this binds with this. This is host, that is human cell, this is virus. This spike protein binds with this host cell and facilitates the entry of the whole virus into the cell. One thing. And second important thing to remember about this spike protein is these proteins are targeted by host neutralizing antibodies. That is the host, when it is recognizing the antigen for the foreign invading material that is coming in contact with the host cell. So it has it is eliciting the membrane the neutralizing antibodies to counteract this antigens. So it is eliciting the host membrane antibodies. and this spike protein is the only part which is targeted by the host neutralizing antibodies. So host neutralizing antibodies are directly targeting this spike protein to evade from the infection. Second is this envelope. So this is like this one. For example, this is envelope protein. Envelope protein interacts with, there is one more protein, this membrane. So envelope is like this. Envelope protein, it interacts with membrane protein or matrix protein to form viral envelope. So viral envelope. This viral envelope protein, that means this one. This envelope protein is important for virus infectivity. So this one is only important for virus infectivity, this envelope protein. So after that we'll see like the third one, which is matrix protein. That's M. This is transmembrane. Matrix protein, I already mentioned, this is transmembrane, that is inside the membrane, not inside, not outside, that's on the membrane surface this matrix protein is on. So it determines the viral shape or viral envelope shape. It determines the shape of it. This matrix protein. Fourth one is nucleocapsid. And nucleocapsid here. So this is the genetic material. Genetic materials, we know that this is single-stranded RNA, right? This is single-stranded RNA here. But over this RNA, there are some proteins like this, like we have in humans, where DNA linked with some proteins. In the same way, here proteins are linked with this RNA molecule, and it becomes the nucleocapsid. This is called nucleocapsid. Nuclear material plus capsid, that means protein. So this nucleocapsid, this binds with RNA genome. This is RNA genome. So this protein binds with RNA genome to make the helical ribonucleoprotein. Ribonucleoprotein means RNA plus RNA is nucleic acid plus protein. So this was about the structure of this coronavirus. And second thing that we need to know about this is the SARS-CoV-2, that is this coronavirus 19. There is one thing on SARS-CoV-2, that is a term in genetics that's called a polymorphism. That is a genetic polymorphism. So there is one important term. So that is genetic polymorphism. That is genetic polymorphism. If we are talking about this S protein, that is spike protein, in SARS-CoV-20, this spike. So when we compare this, when you compare the SARS-CoV-2 with other coronavirus like SARS-CoV or some alpha, some delta, some other coronaviruses, when we are comparing, so there is a genetic polymorphism in this SARS-CoV-2. Genetic polymorphism, so there is some change of some nucleotides in the genetic material of this SARS-CoV-2. This genetic polymorphism of SARS-CoV-2 makes this SARS-CoV-2 a lethal to humans. That is, it causes the lethal human disease such as SARS or MERS. The different spike protein structure. This spike protein structure is very different in this SARS because of this genetic polymorphism. This has suggested that protein can be activated. So there is one more thing that I would like to tell you. There is one thing in alveoli, like in the rest. So I would like to say in lungs, in liver, in small. So in the human tissues like lungs, liver, small intestines, there's the particular enzyme called furin. This furin gets activated by this spike protein. this spike protein is activated by this furin. So furin is actually cell enzyme, host cell enzyme. Importantly, host cell enzyme. So this host cell enzyme furin activates this spike protein of the virus. So where it is located, this spike, this furin, in the tissues of lungs, in liver, in small intestine, and the alveolar lining. So whereas if we are talking about the SARS-CoV and other related coronaviruses, they do not have the furin activation sites. That is, their spike proteins has a structure where there is not the activating sites of furin. So furin is not required for the activation, or they are getting activity without the furin, or furin is not at all needed, or they are not getting activity with the help of furin. So furin is not important in those other viruses, but in COVID-19, furin is important cell enzyme. So this potential furin activation for SARS, that is SARS-CoV-2 or COVID-19, contribute to the manifestations. These contribute to the manifestations of, or we can say they may contribute or expanded cellular tropism, and may contribute to manifestations of liver injury as well. Importantly, so liver injury can be caused by this furin activation of virus. Now my question, that's coming into my mind, that is about how could these specific proteins like spike proteins, membrane protein, envelope protein, or nucleocapsid of COVID-19 be useful targets for diagnosis. So that means how we can diagnose the COVID-19 by looking at either spike protein or matrix protein or envelope protein or nucleoprotein. And the second thing is, what are the molecular diagnosis would be useful? That means molecular diagnosis. We can say useful molecular diagnosis. So we'll talk about regarding this molecular diagnosis and the other targets of that we can how we can diagnose this coronavirus, or how we can diagnose the pathogenicity, or how we can correlate those diagnoses with these spike proteins or the genetic material in the next video. So till then, stay home, stay safe. Thank you.